All right, here I am on our D2L site, and what I'm going to show you today is how to use something called yass. And yass is linked right here on what day is this? Right under Jump to Assignment 4 on August 31st was when I posted this. And I'm going to go to the link here, click on that, and this will be the yass website. You can read about it if you like. You don't have to. You can just watch what I'm doing here. Now, I'm not going to give you an assignment with yass, but when you do your larger project, probably this would help you out. You don't have to use this for the project, but I'm going to warn you that it can save you time. So I'm going to show you how to use it so that you can save some time. Now, what you do, you click on the web server part and you get this. And how you use this is, you remember those GPX genes that we found, those 1 through 8? Well, if you have those somewhere on your computer, you load those in here where it says choose file. There's no file chosen, so you choose file, and then you go find where those files are. And I did save one from somebody's. Let's see, let me see if I can find it here. Here we go. Curtis. Looks like this is Curtis's. So Curtis, thank you for making the file for us. And we're going to load this in here. And we're going to load all those GPX genes in here. And we're going to go select. So you have to hit the select after you do that. And that's going to make it appear down here. You have uploaded file 1. So that's this one. So that is done. Now what you do is you come down here, and you could put some other sequences to compare it to, but what we're going to do is we're going to compare it to an entirely different species. So we're going to take all those GPX genes and we're going to kind of compare them using this database, and we can pick which species we want to do. Now those are 8 examples of human GPXs. So what we're doing here is we're looking for non-human GPXs based on what we know about human GPXs. And so I'm going to go down here to Drosophila, Drosophila melanogaster, or melanogaster, whichever, European or American, whichever way you want to say it. So it's DR, let me find it here. All right, and this one. So this fly is well sequenced. It's one of the best sequenced organisms. Humans, chimps, rats, mice, and flies are among the best sequenced organisms, and you want a really good one here if you're going to do this type of research. So I'm going to choose Drosophila, and then we're going to have to choose something else though. You choose here the chromosomes. Now luckily, unlike us, the Drosophila doesn't have many chromosomes, so we don't have that many choices here. So let's just choose, I've done 2L, I know that one works, but let's do the 2R, and let's just see what happens here. Now, this is a very large piece of DNA, all right, and we're going to be searching for comparison. We're going to be comparing those sequences, the 8 GPXs in humans, to this entire chromosome. It's going to find the places that are similar. So we need to select this now. All right, now you may be using different organisms. Maybe you have to do all the chromosomes or something, depending on the assignment or the project we have in the future. You may have to be comparing to some organism like flies. These are just regular fruit flies, or maybe to mice or to rats or chimps. Those are things that we might be comparing to. And then if you do this, this is going to make your life easier when it comes time to do your project. You can do many sequences at once, kind of like a batch of them all at once, and you have to do them all one at a time. And so we're going to run yass here. Don't worry about the parameters down here, not at this point. I guess we could set a much higher threshold. E value I will explain in another video. Generally, this is about where you want it. I'm just afraid that if I set it that high, this is very sensitive. This means it has to be very close. If I set it that high, we may not get any results. So I'm going to go ahead and set it without very stringent results at all, which were the default at 10. and then maybe we'll go back and do it at a normal setting. So just to show that probably we won't get anything. But let's run this and let's see what we get. Here we go. So what we have here are the, it's still running, hold on. Might take a few minutes. I hope not minutes. There we go. All right, so we can do several things, and if you'll notice, it gives us dot plots. It gives us some tabular data and raw data, and I want to explain BLAST and some other things here. So first, you know what dot plots are. Now these are not going to probably be useful dot plots, at least not without making them zoom, because you're comparing a very small sequence compared to a very large sequence, and it zooms it up and you don't see anything. There'll be little dots and not even visible dots. So if you do it, you're going to want the zoomable one. And here, even with this, like I said, there's nothing here, but if you knew about where it was, you could zoom in. But it makes dot plots, when you do something like a whole chromosome versus smaller sequences, this is pretty much useless. So this is not useful for us, all right? Though it could be if you were comparing smaller sequences. All right, now we're going to go to BLAST data. Let's do FASTA first because you understand FASTA. And then we hit download after that. And what happens is it found sequences that were similar, things that might be lines on a dot plot. So you had that dot plot and you saw a dash on there. Well, that's kind of what you're doing here. And so 1A and 1B, these are the two that were similar. If you were to put these into these two pieces, if you were to cut 1A and 1B out and put it into a FASTA and run it on and compare these two on dot plot, you would see maybe not an exactly solid line. See, there are dashes. Those are pieces that are missing and things. So it wouldn't be solid, but it would be close. And so you go through these and notice you get about a couple of hundred, 82 sequences that lined up. All right, so that's useful to you. Understand how that's useful. This essentially finds all those pieces that we did in the dot plot, but it finds the actual sequences that were aligned, the pieces that matched up. So that's how this is useful to you. So let me go back. The other thing that if you want to do it, that's so you can get the FASTAs, but you can also do a BLAST view. And this is something I'm going to show you how to use BLAST later, but it gives you a slightly different view. You get to go through and see those sequences and see how they line up with the letters there. So essentially these would be the things that would show up on a dot plot as a little line, as a little dash or something. And here you actually get to see the letters when you're looking at them. So this finds all those little places where you would see the dashes. And it gives you the location of them, so from 140 to 199. And see, here's the reason you can't see anything on the dot plot. This one is at, what is this, 23,883,097 to 23,883,038. So they line up right there really well. But this is a tiny, maybe a few hundred bases here, and this one is like millions and millions. So that's why you don't see anything when you do an actual dot plot. And you need to do it this way. And you can actually see the sequence and look at them. And you can get the FASTAs also. So remember, you might want to use these in something else, so you'll need the FASTAs. But when you first look at it, you want to look at it this way and go, oh, look at this. This is interesting. You know, there are some interesting pieces in here, and you may want to visualize it that way. So just realize the ones you're going to want are FASTA and BLAST here. The dot plot, probably when you're using small sequences versus large sequences, aren't going to be usable. All right, so I just wanted to show you this. I won't give you an assignment on yass, but I wanted to show it to you.